Hello everyone, my name is Mobina Kazemi and I am doing the second year of my master's degree in bioinformatics under the supervision of Dr. Jason DeConing at the University of Calgary. My today's presentation is about non-equilibrium molecular evolution caused by fitness shift and drift and how to use DN2DS methods to detect adaptive evolution. Finding a mechanistic molecular basis for adaptive changing function has been always interesting for evolutionary biologists, and since around 1960 they have addressed this question through different approaches and models. One of the earliest models to address the problem was codon substitution models proposed by Goldman and Yang in 1994. This approach models a fraction of non-synonymous substitutions versus synonymous substitutions DN2DS on a phylogeny to provide a sensible measure of natural selection. Throughout time, this model got improved, and the last version of this type, which is branch site model, counts for variable selection concern over sites and branches. Besides all the advantages of these models, codon substitution models in lineage of Goldman and Yang have constant stationary distribution across different sites, and the rate of substitutions from fit to unfit and unfit to fit states are not distinguishable. However, both of these assumptions are not realistic. So, mutation selection models came up as a general solution for these limitations. Here, in our study, we explore how DN2DS changes along a branch of a phylogenetic tree when a mechanistic model of molecular evolution is assumed. In our study, we count for time heterogeneity and let the fitness profile change at any point along the phylogenetic tree. Like here that you can see the fitness profile has changed in this point of a phylogenetic tree. Let's first see how fitness change can happen. Fitness change can happen in two different ways, shift and drift. Fitness shift is when the fitness profile is completely replaced with a new fitness profile, like here that ASG is now PED. Fitness drift, however, is when we have a fitness profile, but the most favorable states are just swapped, like here that ASG now is now SAG. We start from the equilibrium under the first model, and then a switch happens, and it's a non-equilibrium condition that the system is going to reach the equilibrium under the second model. In this situation, we want to compute what are the consequences of that non-equilibrium condition. We are following a simulation-free approach to determine the true DN2DS over time, which is more precise and much faster compared to previous approaches. So, we divide the time frame into small time points and start from the stationary distribution under the first model that has the value of probability of being in each state in this time point. Multiplying this vector to the matrix that has the value of probability of different transitions between different states can give us a new vector that has the value of the probability of being in each state in this new time point. Following this process till reaching the equilibrium under the second model can give us an idea of what's happening in this non-equilibrium condition. The transition probability matrix is the matrix exponential calculated from the derived model rate matrix. Based on this explanation, we can calculate the true DN2DS value at each time point based on this equation. We are focusing on a single branch of a phylogenetic tree, which is going to be representative of what's happening in the phylogenetic context, so we don't need to consider the whole tree. In this study, we used the data from Lee et al. Paper in 2008, which uses a mixture model to infer the fitness profile of 50 site types, and also we added 3 arbitrary profiles to these 50. For our experimental design, shifts can happen between any non-identical pairs of fitness profiles, like fitness profile number 1 can change to fitness profile 2, 3, and so on. A fitness drift can happen for any of those fitness profiles in our input data. The result of fitness shift were categorized in two different groups, those that experience adaptive bursts and those without it. These different behaviors depend on the ancestral and derived fitness profiles as well as the different pathways between codon states. For example, the first panel shows a fitness shift from GND to DNE, and sequences become out of equilibrium with respect to the underlying model, which is typically believed to cause an adaptive burst of non-synonymous substitution, rapidly driving the implied population from having favorable states under the initial model to have favorable states under the derived model. So it would cause an adaptive burst in DN2DS that is shown in this figure. We also represented the frequency of different amino acids over time, and as you can see here, based on the initial state and derived fitness profile, G has decreased, but D and N are increasing. The second panel belongs to the no adaptive burst category, and that's because all the fitted amino acid states in the first profile are still fitted in the second one. The third panel represents the fitness drift, and as you can see, convergence happens very slower for fitness drift compared to fitness shift. For the conclusion of this study, we found out only 61% of the fitness shifts have clear adaptive burst behavior in terms of their time-dependent DNTDS properties. Because our evaluation considered the true nature of the evolutionary process under a mechanistic model of time heterogeneous fitness, they reflect the best-case scenario that any method of inference can hope to achieve. In future work, we will simulate data under a subset of these fitness shift scenarios to directly evaluate when branch-side approaches are able to correctly distinguish true fitness shift from episodic change in population size. Thank you so much for your time.